Welcome back to the screencast for Chapter 1. This screencast, which is number 5, will review the scientific method. The scientific method is really important to our ability to understand and test ideas that we have about the world around us. And it's very important that the process be followed so that we can get data that is worthwhile and we can interpret results which will be more accurate and meaningful. And then we can determine how to accept or not accept the findings. So the scientific method has a series, a process which is always followed. And then within it, there are factors such as in collecting data that may make the process or the method stronger or weaker. Let's walk through this process by first using the case study from the first edition of the textbook. In that case study, there was a place called the Neuse River in North Carolina. And one day, well, actually over a period of a very few days, more than 1 billion fish died in that river. Now that number 1 billion is rather hard to imagine. So again, if you want to refer to the video about conceptualizing billion, please refer to that in Moodle. But I think we can all agree that's a lot of dead fish. So people who go out on boats in the water would see just dead fish floating everywhere. And the question so the observation was, 1 billion dead fish, yikes. The question is, why? So the next step in the scientific method is to develop a hypothesis, a testable prediction as to what might answer the question of what killed 1 billion fish. So one hypothesis might have been that something happened to the pH in the river. Another hypothesis might have been that the water became too warm. Another hypothesis might have been that there was a a disease which caused the death of the fish. So all of these are testable hypotheses. What would not be a hypothesis is something that said, well, aliens came in and killed off 1 billion fish. That's not testable. How would we test whether aliens came in or not? That's not reasonable. So a hypothesis has to be a testable prediction. And for this example, we're going to go with a prediction that a microbe, some microorganism, caused the death of the fish. The next step would be collecting data. And so they went down to this Neuse River and tried to collect data. Was there a microorganism in that water in an amount that should not normally be there? And as an aside, horrifyingly, when the researchers went down to the river, they experienced rashes, nausea, headaches, and other symptoms. But they were there to collect actual data. So they took water samples and looked at the various types of microorganisms that were in the water and compared them to what would normally be in the water. Unfortunately, they are a little bit hampered because there had not been data established prior to this event. So there wasn't a clear baseline to compare. But they could compare the data they collected about microorganisms to other rivers in the same area that did not experience this problem. So when they collected the data, there were different research groups which had different experimental data results. There was a group of researchers who showed that there was a microorganism named Pfiesteria that was present in the water in quantities or concentrations that should not normally be there. And further, they demonstrated that this microorganism, Pfiesteria, went through an unusual life cycle. And part of that life cycle was that when there were high levels of nutrients in the water, it became carnivorous, and it was able to release a toxin which destroyed and ate the flesh of fish. And as a result, there was the data that they had with lots of dead fish. Now, there were also other research groups that did not come to the same conclusions, but we will deal with that during lecture time. So the next step is to interpret the data that you've collected. So let's say you're the researcher, you've collected the data that indicates that this Pfiesteria is the cause of the dead fish. There's a little bit of a human element there to interpreting the results. Then some is clear-cut and some may not be so clear-cut. So the interpreting of results may have a human factor into it. After that, we see whether the hypothesis is supported or not supported. So the hypothesis was that a microorganism caused the death of 1 billion fish. In this particular case, and looking at the particular data that was first obtained, the hypothesis was supported, and a microorganism did appear to have caused the death of the fish. So the next step is to disseminate the findings. Findings may be disseminated through various venues. The best would be through a scientific journal, which is peer-reviewed. That means that the researchers would write up their experiment and their data and interpret their results and send them to a respected journal that would review them, have a panel of people who work in that particular field to review the findings, and if they seem reasonable to publish, then they would publish. Sometimes findings are disseminated in other ways, such as the local media, and that may or may not be a best way of disseminating findings. Obviously, it would get to more people, that's a positive, but findings that are not necessarily peer-reviewed and checking that the scientific method was followed, you may get findings disseminated in the media that are not accurate. You probably have heard things like, 1 cup of coffee is good for you, 2 cups of coffee is bad for you, but 3 cups of coffee is good for you. That doesn't even make any sense, okay? If 1 cup of coffee is good for you, but 2 is bad, how is 3 good? So sometimes when information is disseminated through things like the media, it may or may not be accurate. Sometimes it is. We'll examine this process of the scientific method much more in lab, a lot in lab. For now, I want to move on to some of the factors on the next slide. When data is collected, the value of the data can range from not so good data to really good data. And some of the factors that affect whether data is really good data or really lame data is shown here graphically as a target. So sometimes you can, if this center target represents the area where you have the actual real data, you see that there's a cluster of darts that are closely associated with one another, which might make it look like it's good data, but it's very far off from the actual data. So actual is accuracy. This example here, these darts have low accuracy. They are not close to the actual results, but they have high precision. They all are closely aligned. So that might give you a false sense of security. You get a bunch of data points that all look close together, so you think, oh yay, that's high precision, but it may not be accurate. A second possible scenario is high accuracy but low precision. So here our darts are spread out, but if we average them all out, we would get to high accuracy, high actual results. But they're not precise because they are not closely aligned with one another. The scenario that you would really like to see in your data is high accuracy and high precision. So it's high accuracy that if you average these out, you'll get to the actual result, and high precision because the darts are closely aligned with one another. Okay, so that is looking at data. I'd like to take a minute now to look at some scientific terms, and then we'll go back to data collection. Okay, here are some scientific terms that you need to understand. We have the term theory and we have the term natural law. The term theory is not used in the same way or defined in the same way in science as it is in regular language. In regular language, you might say, I have a theory of who committed the crime. That means a guess. That's not the definition in science. In science, a theory is a hypothesis, an estimate, that has been, and here's the difference, repeatedly tested and confirmed by multiple groups of researchers and is widely accepted. For example, is atomic theory. So if you've ever watched The Big Bang or studied a little chemistry, you might know that atomic theory basically says that matter is made of individual atoms. And these atoms contain three main parts: a nucleus, which is comprised of protons and neutrons, and an outer shell, which comprises electrons. Now, that hypothesis has been repeatedly tested and confirmed by multiple groups of researchers without actually even visually seeing an atom. And because it's a theory, it means it's pretty close to probably what is, but it doesn't mean there couldn't be exceptions or more information basically that you would find about atomic theory. So in fact, over the years since atomic theory was first developed with the three main parts of an atom and then the three main parts of an atom, it has been tested and confirmed by multiple groups and researchers that atoms are actually comprised of multiple subatomic particles. So not just the protons, neutrons, and electrons. So it didn't change the overall theory that an atom is the smallest unit of matter, but the actual details can change. A difference here, another term, is natural law. So that's when a theory has been tested multiple times and there are no known exceptions, and we don't think there ever will be. So an example here, I like the law of gravity. The law of gravity. So if you go up and you go up to a tall tower and a skyscraper, and you have a basketball and a bowling ball, and you drop them at the same time from the same height. Which one will hit the ground first? The basketball, the bowling ball, or will both reach at the same time? Well, both will reach at the same time. And that has been tested all over the world in many different environments and explained because it's the Earth's pull on the object, not the object's falling, as we might think of it, to the Earth. So that's been tested and tested and tested, and there are no known exceptions, and there are not expected to be any known exceptions. So it becomes a natural law. At this point, we're close to the end of the 15 minutes, and I do have more to say about the scientific process, so I'm going to end this video now, or the screencast now, and move on to our final one.